Good afternoon, everyone. My name is Revathi Manoharaan. I'm from the University of Illinois, Urbana-Champaign, and today I'm going to be talking to you about unveiling hypoxia patterns in a breast cancer progression model. So, tumors often start with a few cells dividing uncontrollably, accumulating mutations up until to the point that they form a huge mass of cells like this. Here's where the problem starts. Because of the tumor being so big, there is a lack of oxygen penetration, and this leads to a condition called hypoxia, where some of the cells inside don't get enough oxygen. You might think this might be a good thing, right? Tumors will probably die if they don't have enough oxygen, but no, this becomes the reason why more resistant cell lines become selected for, and therefore leads to tumor aggressiveness, resistance to therapy, and therefore patient mortality. This unfortunately affects 40% of people with breast cancers and particularly is common in triple-negative breast cancer, which is a much more aggressive form of breast cancer affecting a huge number of people and is not responsive to hormone-based therapy. Today, we are going to be using this triple-negative breast cancer model called the MCF 1080 series, which consists of 4 different cell lines starting from normal all the way to metastatic carcinoma, and we're going to subject these to hypoxia to see how they respond and characterize that. So, how we're going to do that is to first characterize them in terms of their chemistry and then their morphology, followed by their spatial distribution. What we're going to do is to grow these cells in 2D and then subject them to different levels of hypoxia and then analyze them using IR spectroscopy to look for chemical biomarkers and then quantitative phase imaging to look for morphological changes. For FTIR imaging, what we do is to illuminate the sample with the IR-based beam, and then each of the chemical bonds in this sample have different types of vibrational modes and hence absorb different wave numbers of IR light. What we do is to plot these absorbance at different wave numbers to form an IR spectra like this at each pixel so that we can get an image. And each of these peaks actually correspond to different types of biomolecules. So what I'm going to present to you today is the cell average FTIR spectra for the different samples. And we start out with the region that is representative of nucleic acid and polysaccharides. For M1 and M2 cells here, what we see is that there is an increase in there is an emergence of new peaks at the DNA region, and these are often associated with DNA structural changes and possibly DNA damage. When we looked at the area under the curve for these peaks, we also saw that there was a significant downregulation in nucleic acid content, especially in M3 and M4. Furthermore, when we looked at these polysaccharide peaks, we see that there is an upregulation of polysaccharide peaks and emergence of these 3 main peaks, which are representative of glycogen in M3 and M4. Next up, we looked at the protein, and we look at the shifts in these protein peaks, amide1 and amide2 peaks, to look at what kind of secondary structural changes happen in these cells in response to hypoxia. So M1 and M2 showed a rightward shift, whereas M4 showed a leftward shift, whereas M3 remained fairly constant. So this suggests that M1 and M2 are probably undergoing an increase in alpha helical structures, whereas for M4 there's an increase in beta sheets and aggregates, whereas M3 remains fairly constant, which suggests that maybe they are already adapted in terms of their protein structure to survive hypoxia. As for the lipids, we saw that there was a general upregulation overall in all the cells in response to hypoxia, and particularly there was a progressive increase from M1 to M3, whereas in M4 we saw a very slight increase over time. So this gives us a very wide range of chemical biomarkers to work with, and next up we're going to look at the morphological changes. What we did was to analyze these samples using spatial light interference microscopy, which is a type of phase imaging that is quantitative, which allows us to look at the changes in the refractive index of the sample. So when we looked at M1, we saw that there was the shape of the cell significantly changed to become more circular and more spread out. As for M2, along with this morphological change, we also see the emergence of these lipid droplet-like structures, which are high refractive index droplets inside the sample. And in M3, we saw even higher upregulation of these lipid-like droplets. As for M4, we see that it already had a lot of lipid-like droplets, so there wasn't much of a change over time. And this was actually verified that these are indeed lipid droplets based on fluorescence microscopy. Other than that, we also found that there was an overall increase in the face value of these samples, probably because of the high refractive index lipid droplets that are appearing in these samples in response to hypoxia, along with wider cells and circular cells. And then the next part of the study is where we grew these cells in 3D, where we co-cultured fluorescently tagged M1 cells with M3, M4, and M2 to M4 cells, and then we grew them to form tumors that were more than 200 microns to create an oxygen gradient and see how they will spatially arrange themselves. Here are the results, where we started off with M1 and M2, where most of the M2 cells were in the periphery and M1 cells were in the center, mostly dying. As for M3, the M3 cells dominated the hypoxic core, whereas very few M1 cells were on the surface, and M4 cells showed a rather heterogeneous mixture. With that, I have derived the hypothetical arrangement of the different cells in response to hypoxia, and we're hoping to combine the results of this hypothetical arrangement plus the morphological changes and the chemical signatures that we have identified to be able to bridge the gap in label-free tissue imaging, tissue hypoxia imaging. With that, I'd like to conclude my presentation. Thank you.